You know, there's that old phrase in tech that everyone loves to quote, software is eating the world.
A classic.
It is. But if you look at what's happened in, just the last few months, this transition from late 2025 into early 2026,
Mhm.
it feels like software got bored of eating the world and decided to start rewriting biology instead.
It's a massive transition point. I was thinking about this the other day. For the last, 50 years, biology has been a descriptive science.
Right.
We were archaeologists,
Yeah.
brushing dirt off bones, trying to guess how the dinosaur walked.
From a fossil, yeah.
Now we are effectively cloning the dinosaur. Or better yet, we're designing a dragon because, the dinosaur didn't have the features we needed.
That is a simultaneously terrifying and exciting image.
It is.
But it really captures the shift we're seeing. We're moving from just reading the code of life to actually writing it.
And that brings us to today's deep dive. We are unpacking what we're calling the era of generative biology. So specifically, we're looking at a stack of 4 papers that have effectively dropped a new toolkit on the desk of every drug hunter on the planet.
And these aren't just theoretical papers that might matter in 10 years.
No, not at all. These are tools that are live or coming online right now.
And this isn't, AI got slightly better at spotting a cat.
Mhm.
This is a complete pipeline overhaul.
Right.
We're talking about the new AlphaFold Database 2025 update, which changes the map we use.
Then AlSites, which tells us where to aim on that map.
O-Design, which is basically a world builder for molecules.
And finally, OMTRA, which refines those designs into actual functional drugs.
Right. Think of it as a 4-step assembly line.
Exactly. You need the blueprint, you need the target coordinates, the fabrication tool, and then quality control. These 4 papers are those 4 steps modernized.
Okay, let's unpack this. We have to start with step 1, the blueprint. For the last few years, if you said protein structure, everyone just shouted AlphaFold. It felt like the problem was solved. We all pack up, went home, job done.
That's the common misconception. AlphaFold 2 was a breakthrough, no doubt. It gave us the shape of almost every known protein. But it had a significant blind spot.
Which was?
It was static. It gave you the standard version of a protein, the canonical sequence.
The factory setting, the Honda Civic as it rolls off the line.
Exactly. But biology doesn't run on factory settings. Our genes are constantly splicing and dicing RNA to create variations of proteins, which we call isoforms. So one gene might produce 5 or 6 different versions of a protein, each with a slightly different shape, a different tail, maybe a missing loop.
So if AlphaFold was showing us the standard Honda Civic, the biology inside a patient might be driving a Civic Type R, or maybe one that's been modified into a boat.
Or a Civic with a flat tire and no steering wheel. And this matters immensely because disease often hides in those modifications.
Oh, I see.
If you're designing a drug to fit the ignition of the standard version, but the cancer is driven by the weird isoform that uses a push-button start, your drug fails.
You're holding the wrong key.
Precisely. The AlphaFold Database 2025 update is massive because it finally syncs with the UniProt 2025-03 release. It brings in these isoforms.
So it's the director's cut of the proteome.
Yes. That's a great way to put it. And it's not just about listing them, it's about structure and function. The update integrates something called TED.
Which I assume is not the conference with the red carpet.
No, not that, TED. This is the Encyclopedia of Domains.
That sounds like a very dense fantasy novel.
Well, it's actually thrilling if you're a structural biologist. Domains are the functional units of a protein, the engine, the wheels, the steering mechanism.
Okay.
TED adds annotations for 361 million domains across 165 million proteins. And crucially, they added a new metric called Q-score.
Which is what? A Yelp review for protein quality?
Pretty much. It tells you how confident the AI is about that specific domain assignment. Before, you just had PLDDT, which was an overall confidence score.
The whole thing.
Q-score is much more granular. It tells you, I'm 99% sure this is a kinase domain, or I think this might be a binding site, but, don't bet the farm on it.
That sounds like a massive quality of life upgrade for anyone trying to navigate these huge data sets. And I saw they overhauled the visualizer too.
Yes. The visualizer was, let's say, functional before. Now it's an analytical tool. You can overlay the domain data directly on the 3D model.
So you can actually see the different parts.
You can toggle between the confidence view and the domain view right there on the model. It turns the database from a reference library into a real workspace.
So step 1 is complete. We have a high-definition, up-to-date map with all the weird alternate versions of proteins. But here's the next problem. Having a map of a city doesn't tell you where to park the car. You have this complex 3D shape. where do you stick the drug?
This is where we tackle the classic binding site problem. And this leads us to the second paper in our stack, all sites.
Right, and just to give some context for you listening, usually if I want to find where a drug binds, I need a specific tool for a specific job.
It's incredibly fragmented. If you're looking for a pocket where a small molecule fits, you use one tool. If you want to see where DNA sticks to a protein, that's another tool.
It's siloed.
Totally siloed and slow. And historically, you needed the 3D structure first. You had to run AlphaFold, get the shape, and then scan it. AlphaFold flips this entire logic. It says, I don't need the structure.
Wait, what?
Just give me the sequence.
It predicts the 3D binding site just from the 1D list of letters.
Yes. It uses a protein language model, ESM2 specifically. It treats the sequence like a sentence, extracts the meaning or embeddings, and then pipes that through a gated convolutional network.
Okay, hold on. Gated convolutional network. Why do we care about that specific bit of jargon?
Because it's great at understanding local context. In a protein, a binding site is formed by amino acids that might be far apart in the sequence, like word 5 and word 500.
But when it folds up,
they end up right next to each other. The network, combined with a transformer decoder, figures out these spatial relationships without building the 3D model first.
which makes it
Blisteringly fast. We need to pause on the numbers here because they are absurd.
okay.
Traditional methods can take minutes or even hours per protein. All L sites screened the entire human proteome, over 20,420 proteins, in just 16 hours.
That's what, 2-3 seconds per protein?
2.81 seconds on a single A100 GPU.
That just changes the whole logic of discovery. You can stop saying, let's study this one protein for a month, and start saying, scan the whole human body.
Precisely. And find every protein that has a pocket for this kind of drug. You can screen for side effects before you even make the molecule. But the real insight here is the universality, hence the name AlSites.
Doesn't care what binds, right?
No. It predicts sites for DNA, RNA, peptides, small molecules, and even carbohydrates.
And carbohydrates are usually the nightmare scenario for computational biology.
They are. They're tricky because they rely heavily on these hydroxyl groups, and standard geometry tools often miss them completely. AlSites picks them up. This opens the door to the undruggable proteome.
We hear undruggable a lot. It usually just means we can't find a pocket for a pill.
Right. But AlSites might look at an undruggable cancer target and say, okay, sure, no pocket for a small molecule. But look right here. This surface is perfect for an RNA aptamer. It expands the whole menu.
So we have the map from AlphaFold. We've used AlSites to find a hidden landing site. Now comes the part that feels like actual magic. We need to design the thing that lands there. This is where we talk about O-design.
This is the heavyweight of the stack. The authors call it a world model for biomolecular interaction design.
World model is a heavy term. In AI, that implies it understands the physics and rules of the environment, not just pattern matching.
And that's the claim here. O-design isn't just a protein generator, it's an interaction generator. Most tools we've seen, like RF diffusion, are protein-centric.
Right, protein in, protein out.
O-design is all-to-all. Meaning you can input a protein, a strand of DNA, a piece of RNA, a small molecule, or even a metal ion, and ask O-design to build a binder for it.
How does it speak all those different languages? A DNA helix and a protein sheet are totally different.
That's the innovation. They created a unified token space. They basically abstracted the chemistry, amino acids, nucleotides, atoms into a shared language.
I see.
So to the AI, a carbon atom in a drug and an alanine in a protein are just different tokens in the same vocabulary.
And that lets it read the interface between them.
Correct. And it uses a diffusion model, similar to how image generators create pictures from noise, but the killer feature is the flexible receptor mode.
Explain that. Why do we need flexibility?
Because the lock and key analogy from high school biology is, well, it's a lie.
It's a useful lie.
A useful lie, but a lie. Proteins aren't stiff blocks. They're like memory foam. When a drug hits them, they shift. They mold around it.
Induced fit.
Exactly. O-design allows the target and the binder to co-evolve during the generation process. It wiggles the protein while it designs the drug.
Let's look at the scoreboard, because the throughput comparison against RF diffusion was startling.
RF diffusion is the current celebrity in this space. It's the benchmark. In a protein binding task, it could generate maybe 555 successful designs per day on a GPU.
And O-design.
2,672.
So 5 times faster.
And orders of magnitude more versatile. They tested it on a zero-shot task-designing RNA aptamers, which are notoriously floppy and hard to predict for proteins that it had never seen. It hit a 77.95% success rate.
That is just unheard of.
It is.
There was one other feature in O-Design that caught my eye, tip atom scaffolding.
Oh, this is cool. Imagine you want to build an enzyme that cuts a specific chemical bond. You know exactly where the 3 atoms of the blade need to be in 3D space, but you have nothing to hold them there.
You have the knife blade, but no handle.
Exactly. O-Design can take those few atoms, the motif, and build a massive functional protein structure around them to hold them in that exact configuration.
It's like building a cathedral just to hold a single candle in the perfect spot.
A very functional cathedral. It moves us from finding drugs to programming them.
Okay, so O-Design is the architect. It creates the new protein or RNA. But sometimes you just want a classic pill-in-a-bottle small molecule, and it has to be perfect. That leads us to the final paper, OMTRA.
OMTRA is the specialist. It's a structure-based drug design, or SBDD, engine.
And the big shift here seems to be moving from diffusion to something called flow matching. In simple English, what's the difference?
Think of diffusion as starting with a cloud of noise and slowly unblurring it until it looks like a molecule. It works, but it's chaotic. Flow matching is more like drawing a direct line from the noise to the final shape.
More efficient.
Mathematically, yes. Fewer steps. But reading the paper, the real secret sauce here isn't just the math, it's the pharmacophores.
Yes, let's define that for everyone. What is a pharmacophore?
It's the ghost of the interaction. It's an abstraction. Instead of saying, put a carbon atom at coordinate X, a pharmacophore says, I need a positive charge in this area and a greasy hydrophobic ring over there.
It's the vibe check of the molecule, the functional requirement.
Exactly. OMTRA allows for pharmacophore conditioning. You can tell the AI, design me a drug for this pocket, but it is mandatory that it makes a hydrogen bond at this specific point.
So you're constraining its creativity.
You're guiding it. And the data shows this is crucial. Adding just 3 of those constraints reduced the sampling steps needed by 56%. It stops the AI from exploring useless chemistry.
Now, to train an AI to understand chemistry this well, you need data. And the bottleneck is always data.
Always. So the OMTRA team did something incredibly clever. They built a synthetic dataset called PharmIt. They generated 3D shapes for 500 million molecules.
500 million.
It's one of the largest open-source collections of 3D molecules ever created. This teaches the model chemical plausibility.
Which is the AI knowing that atoms can't overlap and bonds can't be 10 feet long.
Right. It separates the problem. First, learn what a valid, stable molecule looks like from PharmIt. Then, learn how to fit that molecule into a protein pocket. If you try to do both at once with limited data, you get garbage.
And they tested this with pose busters, which is a physics check, right?
Yeah, it checks if the molecule breaks the laws of physics. OMTRA hit 91% validity on pose busters. It actually beat AlphaFold 3 on some specific redocking benchmarks.
And it handles the messy stuff, the NPNDEs.
Non-protein, non-designable entities.
That is possibly the worst acronym of the day.
It's terrible, but it refers to things like water molecules, ions, cofactors that hang out in the binding pocket.
Ah, right.
Most tools ignore them. OMTRA sees them and designs around them.
Okay, let's zoom out. We've gone through 4 dense papers. Let's construct the pipeline for everyone listening. How do these fit together tomorrow morning in a lab?
Okay, step 1. You have a disease target. You go to the AlphaFold Database 2025. You look at the specific isoform active in your patient's tissue. You use the TED annotations.
You get the specific director's cut map. Step 2.
You take that structure, or even just the sequence, and run it through all us sites. In seconds, it finds a hidden pocket that traditional tools missed.
Step 3.
You make a choice. If you want a large biologic, a protein or RNA, you plug the coordinates into O-Design. You let it co-evolve the binder and the target.
And step 4.
If you decide you want a small molecule pill, you go to OMTRA. You identify the key interaction points, the pharmacophores, and you force the model to hit them.
It feels like the element of luck is being systematically removed from the equation.
That's the goal. We're replacing serendipity with engineering. But there is a final thought here, something O-Design hints at that goes beyond just making drugs.
The all-to-all capability.
Yes. If O-Design can design a protein that binds to a specific sequence of DNA, or an RNA that binds to a specific metabolite, we aren't just making therapies anymore.
What are you doing?
We are building logic gates.
Explain that.
A protein that binds to DNA can turn a gene off. An RNA that binds to a small molecule can act as a sensor. If you can design these interactions at will, You can build synthetic regulatory circuits inside a living cell.
You could program a cell to detect cancer and release a toxin, but only if condition X and condition Y are met.
Exactly. We are approaching the era of cellular programming. These tools, the map, the site finder, the world model, and the refiner, are the compiler for that code.
We're not just writing drugs, we're writing the operating system.
That is the horizon.
Well, on that mildly existential but incredibly hopeful note, we're going to wrap up.
It's a good place to stop.
If you want to play around with the new AlphaFold viewer or see just how many domains exist in your favorite protein, we've linked the Encyclopedia of Domains and all 4 papers in the show notes.
Definitely worth a look, even just for the visuals.
Thanks for joining us on this deep dive into the generative biology stack. It's a brave new world out there.
Indeed it is.
Until next time, keep learning.